Welcome. Hi, everybody. Thanks for coming to Chestnut Chat again. I see a lot of regulars on here. I'm Sarah Fitzsimmons. I'm the Chief Conservation Officer for the American Chestnut Foundation. I'm coming to you from West Lafayette, Indiana today, and we have the Indiana chapter meeting tomorrow. So excited to see some of you in person today and tomorrow. Housekeeping Q&A. If you have a question, please use the question and answer or Q&A module. That's going to be a lot easier for us to keep things coordinated and get the questions answered. I want to thank the RSCs and MetaView staff on here, Hoy, Kendra, Jamie, Sierra, Cassie, Lauren, for answering a lot of those questions. We'll also save some of them at the end for live questions. If you just want to do some witty banter, throw that in the chat. Today, we're going to talk about some of the exciting new developments that we're working on, continuing to work on, or looking to in the future to work on with TACF science. We're going to go over some traditional breeding methods that we have ongoing and improving that material. Jared Westbrook is not able to be with us today live. He's on an airplane right now, but he recorded his session, so we'll be playing that. Actually, Kendra will. Jason Holliday's here. Hey, Jason, thanks for joining us. And we'll be talking about the work to find resistance within Castanea and Cisgenics and things like that. Vasily Lakoba is here from MetaView TACF staff to talk about sort of the overview of where TACF science is going. And Bruce Levine is here. He's at the University of Maryland and very active in our Maryland chapter to talk a little bit about what we're doing with the fungus. And then Scott Merkel, same, could not be here live, but he recorded his session. So I want to thank Kendra for running the slides today. My connection here in Indiana is not quite that stable. So Kendra, if you don't mind going ahead and getting that shared, we'll get started. All right, thanks, Kendra. So this is about the future of TACF's science programming. Just a few pictures here. These are pictures that you'll see on into our program today. So I love these two trees that are flanking either side of here. Kendra, I think this is a tree up in Maine. There's Eric there. So this is one of the trees from our back crossbreeding program, and then Scrivani there on the left with a tree from Lasagne State Forest. We also see some of the trees from Scott's program there, some of the newest trees that he's creating there at the University of Georgia. Next slide. And I want to bring up a slide that we presented back in September. And I think there's been a lot of concern and question about Darling 58 and what TACF is going to do next with the program moving forward. And I want to remind that restoration is a process and not a product. So just because one particular product is not going to be the end-all be-all for American chestnut restoration, that was really never the goal anyway, because restoration is going to be an iterative process. It's going to take time to develop. And material that's already out in the landscape, especially material, so hybridized material or backcross material, Jared's going to cover that here in a minute. We have some really great materials that are already out in the forest that are already contributing some ecosystem services to the wild populations that are out there. And over time, as we develop new products like the ones we're going to talk about today, those will then augment material that's already out there and improve those ecosystem services that are already being out there and provided to the greater ecosystem. So you've got the research wing, you've got the implementation wing. You're always assessing, you're always improving, you're always evaluating. But restoration really is a decades, if not centuries long process, allowing us to make those improvements over time. Next slide. And then just to go back and kind of see what we've built on in the past, a lot of this material that we're working on currently and what we'll be creating in the future stands on the shoulders of work that's been done in the past. So, we all are aware that the chestnut blight was brought over in the late 1800s, and before that, Phytophthora root rot was brought over. So a lot of the work that we're working on today is because people had some foresight to say, look, this is an issue. We need to start doing some work to try and recover the what we're losing with this species, with the American chestnut. Obviously, none of these products or programs worked full for a long period of time, but everything that we do augments what had been done in the past, and that's what's going to happen in the future. All of these products, all of these programs will augment and further improve what we have already on the landscape, and eventually see restoration. So that's what we're going to talk about today. Some of these techniques that are ongoing, and some of the future things that we're looking to produce in the future.
All right, thank you, Sarah. So I'd like to set up for all of you kind of the landscape within which we find ourselves when we want to do something as complex as restore the American chestnut tree. So zooming out a little bit, it's important to understand that because this is a very complex problem, bridging multiple scientific fields, bridging, large timescales, that there are a lot of questions. It's a landscape full of questions. Some of these questions are about some basic scientific points of interest, like what types of gene networks confer disease resistance, for example, to chestnut blight. What kind of networks also confer resistance to Phytophthora root rot resistance? What kind of resistance can be combined for those two diseases, both of which are, important throughout the eastern U.S. With some other questions related to the genetics. For example, how can we alter gene expression as opposed to just the genome at a base level to make change? What is the simplest and most compact genetic change that can be made that can lead to success? Some of these questions starting to get more applied in terms of manipulating what we learn from the basic science of the genetics of disease resistance. But then we also want to look at these broader questions of what does it really take, on what time scales, on what spatial scales, to evaluate performance in terms of disease resistance of trees, in terms of their functions in the ecosystem. How can we maximize those benefits to ecosystems that Sarah was mentioning? And also at what time scales can we make reliable predictions about how they will actually perform? Then again, some basic scientific questions about biochemical signaling, and then new tools that are becoming available with each coming year that allow us to look at that in a more sophisticated way and find what are the most impactful changes that can be made. And of course, there are questions of stability, of resistance, any kind of conferred resistance over time, because of course, trees are long-lived organisms. The benefits they confer to ecosystems also happen over large timescales. It's very important. And then, of course, the questions beyond science in terms of regulatory success of different approaches that require genetic modification. And there's many questions beyond these. These are just some of these. But it's very important that we acknowledge that there's a complex landscape of these big questions, and many of them have not been addressed yet in full. Certainly some are being looked at kind of a little bit in their infancy. Others, we're already a significant amount of the way along this path. And some of those our speakers will be presenting on today. But again, it's important to see a very complex landscape of many questions, all of which deserve many years of work. And it's important to understand that there will be no silver bullet solution that magically solves all of them, but a number of approaches taken in parallel is what is most likely to get us to success at diversifying our efforts. So again, understanding that when you have a diverse set of questions, all of which are complex, coming from different scientific and other fields, and we want to make sure that we don't just rely on narrow answers to narrow questions. It's very important to have your eggs in multiple baskets, have diversified scientific approaches, diversified applied approaches, and it's important to understand that a lot of the answers come on tree time. So trees have to grow. You have to evaluate things at the rate at which trees grow, at the rate at which they perform. So having diversified parallel scientific efforts and being patient with tree time really is the recipe for success and that will allow us to get there efficiently and in a way that looks at the situation from all angles. So just a preview of the different talks that you'll be hearing today as part of this Chestnut Chat. So immediately following this, you will hear Jared Westbrook from TACF talking about the ongoing improvements to traditional breeding, so building on existing foundations in that regard. Scott Merkel from University of Georgia is going to speak about new transgenic lines being developed and those projects underway. Jason Holliday from Virginia Tech was going to talk about some of the latest in terms of discovering resistance genes in other Castanea. And then also Bruce Levine is going to talk to us from a plant pathology perspective and some new ways of looking at plant pathogen interaction. And that's going to be very interesting as well. And finally, Kendra Collins is going to close it out with talking about the most important citizen science contributions that'll be coming up in the next years and into this decade, and how all of that fits together into actionable steps for our volunteers and chapters. So without further ado, next slide, and we'll turn it over to Jared.
Hi, I'm Jared Westbrook, a geneticist with the American Chestnut Foundation. Today I'm going to be talking to you about a plan for making traditional breeding work better. You're going to hear about other approaches to improve disease resistance, and by no means is this our only approach to improving blight resistance in American chestnut, but I just wanted to highlight a few advantages of this approach. First, there's no need for federal regulatory approval, in contrast with anything that we do with biotechnology and transgenics. So that means that everyone can participate, and substantial gains in disease resistance are possible, but we don't need to know the underlying genes. So we can make gains without knowing the genes just by selecting the best parents and further improving the progeny. So the plan basically is, step one, we need to select good parents. That's the very important first step in making gains in resistance. We breed these best parents together, select better kids, and now we're using genomic testing to be able to make those selections and plant the next generation seed orchards. So in order to this first step, select good parents, we've been going around over the last few years and looking at the long-term blight resistance of the backcross trees in over 100 locations across the range that have been maintained by TACF's chapters. And we go through and we look at each tree and we assess whether the main stem is alive, whether or not the tree has large cankers, whether or not the cankers are sunken, swollen, percent canopy of the tree that's alive, the presence or absence of exposed wood, the fungus, whether it's sporulating and reproducing on the stem, and whether the tree is resprouting from the roots. All of these are indicators of whether a tree is susceptible to blight. And so we're looking for trees that have largely the absence of these traits in order to make selections. And so we, sum all of those traits together into an index, a blight resistance index, which I scaled from 100 being the average resistance of Chinese chestnut to 0 being the average resistance of American chestnut. And basically we're aiming to select trees, which I call blight 1 selections, that have resistance greater than 40 on that 0 to 100 scale, and American chestnut ancestry greater than 60%. So we're selecting for intermediate resistance and a majority American chestnut ancestry for our best parents. We also have, there's about 100 of these so-called blight 1 selections, and we have hundreds more of blight 2 selections, which have resistance 25 to 40 on that resistance scale. And so those are backup trees in case we need those and the blight 1 selections are not flowering. So this is kind of an example of what these trees look like. Many of our chapters have, a few of these trees at multiple orchard locations. And so this is just showing some of our volunteers that have worked on the program over the decades with these best trees. They're often one tree in an orchard, and their other trees around them are not as resistant. So it's important to, rather than just collecting seed from these individual trees, to be able to make controlled pollinations between good moms and good dads from different orchard locations. So in order to do that, we've been freezing pollen from these selected parents. So when they're flowering in the summer, We collect pollen, we send it off to Meadowview, Penn State. They are able to freeze the pollen and keep it available for future years for breeding. So we've been starting to collect kind of a library of different best parents, their pollen, and then we ship it out to various orchard locations where people are able to do controlled pollinations with the best parents' pollen onto mothers that are also very resistant. Wherever possible, we've been using large surviving American chestnuts as parents in the breeding program. These trees have partial resistance to blight. So they tend to have this kind of cruddy bark phenomenon where they get blight up and down the stem, but it's superficial and it's not girdling the tree. So the tree is able to survive. And here are a few examples, pretty spectacular ones of trees that have lived, even through the original blight epidemic. This is the Ort tree on the left, the Walbridge tree in the middle. These are two trees from Pennsylvania, and then the Amherst tree in Virginia. So we've been crossing with those, and we can definitely improve the American chestnut ancestry without sacrificing blight resistance by using these trees as parents. So the third step is selecting better kids. And how we're doing that now is we can plant these progeny out in a greenhouse, keep track of who their mom and dad is with these tags. We've been collecting leaves into 96 well plates from these trees. We basically take a little hole punch and collect them down into a small little 96 well plate. And we can send those leaf samples off for genotyping. And we're starting to work with this company called Diversity Arrays Technology in Australia. And they can do the genotyping for us for a cost of about $20 a tree. And through that genotyping, they'll send us back the data, and then we'll be able to assess the American chestnut ancestry and the blight resistance from the DNA sequence and make selections of better kids. And the aim is to select 10% of the progeny that have greater resistance than their parents. One advantage of the genotyping, another advantage is not only can we predict blight resistance, we can also predict resistance to Phytophthora root rot, which is another devastating disease of American chestnut. It's been especially devastating in the South recently, where whole orchards have succumbed to this root rot disease. And some of our hybrids have resistance, and we're incorporating that resistance into the program through selecting those trees and breeding them with our blight-resistant selections. And then, of course, we need the trees to be competitive, and we can predict basically through the DNA how tall the trees will grow at early seedling stage. So that enables us to select for these multiple traits that are really important for the tree's ultimate restoration worthiness at one time at the seedling stage. And then we can plant our plan is to plant these next generation seed orchards with the best progeny from the best parents, and plant these progeny out at optimal spacing, and wait about 7 to 10 years for them to produce seed. And a major advantage is we don't need to inoculate the trees. We've already selected for blight resistance through the genotyping. So ultimately, the trees are going to remain healthier for a longer period of time, hopefully produce a lot more seed, and we can start to plant those out in restoration trials. So what to expect from this? I've done some simulations looking at predicting what is the average blight resistance and American chestnut ancestry from doing these best by best crosses and then selecting best kids through the genomic selection process. And basically this is looking at blight resistance here on the top panel. And you can see there's a lot of variation in blight resistance from 0 all the way up to above 80 here. And a lot of those trees are high at Chinese ancestry. And so our average resistance right now is about 10 on that 0 to 100 scale for the whole program. But if we were to select these few best parents and cross them together, we can increase the resistance up to about 44 as the average resistance of the population. Some trees will have more resistance up to 66. Some trees will have less resistance. And that variation is what we can use to actually select for and improve resistance in future generations. So if we were to do a second generation of selection, we could improve the resistance up to about 53 on this scale, and the maximum resistance would increase to about 78. So there is great potential to improve the resistance. And with the genomic prediction and selection for, we can also select for American chestnut ancestry. So we're not expecting to necessarily increase the Chinese chestnut ancestry by way of doing the selection for blight resistance. We're expecting the average Chinese or American chestnut ancestry to remain around 80% over 2 generations of selection. So that, the genotyping enables us to select for greater resistance without necessarily reducing the American chestnut ancestry of the trees. So there's great, potential in, incorporating these really stringent parent selections and the genomic prediction to definitely make large gains in blight resistance. So please work with us on implementing this new exciting plan for the breeding.
Hi, I'm Scott Merkel at the University of Georgia's Warnell School of Forestry and Natural Resources. My lab's been working on chestnut in vitro propagation and gene transfer for about the last 35 years. I want to briefly tell you about some new transgenic approaches for generating blight resistance in American chestnut that we are working on with Jared and other TACF scientists and collaborators. Some involve transforming new American chestnut culture lines with the OXO gene, but we're also looking at other approaches. For the new transformation work, we are using a set of American chestnut cultures called the founder lines. So what are these founder lines? They are new American chestnut embryogenic culture lines from source trees, mainly large surviving American trees, representing different parts of the American chestnut range. We've already screened the new lines and picked the best ones to transform to produce the transgenic trees. These 2 photos here show newly initiated founder lines, and you can actually see the seed that the culture has proliferated from in each of these photos. And this bottom photo shows some trees were generated from one of the founder lines, one that came from a Pennsylvania tree. And this is how we screen these lines to make sure that before we transform the cultures, we could regenerate trees from them. This flowchart shows the protocol we developed at UGA, and we've used it to produce transgenic chestnuts with a variety of genes that we wanted to test over the past 15 years. Slide shows the production of chestnuts engineered with the GUS reporter gene, which makes an enzyme that we can easily detect with an assay that produces a blue stain. Our use of these embryogenic suspension cultures for this process makes it a very powerful and scalable way to make transgenic trees. Even so, it is a very painstaking process, and it requires about 2 years to go from the transformation to potted transgenic trees. So before we go any further, I need to define some terms specifically regarding gene promoters. So what is a gene promoter? A gene promoter is a short DNA sequence just upstream of a gene that turns the gene on. There are different kinds of gene promoters. Constitutive promoters are promoters that keep a gene turned on all the time in all the tissues of a plant. But there are other kinds of promoters. Inducible promoters only switch a gene on with a certain stimulus. For example, a wound in the bark of the tree might turn the gene on. There are also tissue-specific promoters. These only turn a gene on in specific tissues of the plant. Now, I should tell you that the promoter that drives the OXO gene in the Darling 58 tree is a constitutive promoter, but it doesn't come from a plant. It comes from a virus. Specifically, it comes from a gene in the cauliflower mosaic virus. So it's a viral promoter, not a plant promoter. So the work on our first new transgenic approach is already underway. Our approach here still makes use of the OXO gene, but instead of a viral promoter driving the OXO gene, we want to use a plant gene promoter. We believe that a plant gene constitutive promoter may help the plant better modulate the expression of the gene, causing fewer undesirable effects than using a viral gene promoter. The specific promoter we're using is from the Arabidopsis plant, and there's a gene that's always on in most plants, actually, called the ubiquitin gene. And the ubiquitin promoter is a constitutive promoter, but it's a plant constitutive promoter. So if we use that, we hope that we can better adjust the expression of the gene so that we get the just right temperature of the oatmeal here, like Goldilocks likes, or the just right expression of the gene in chestnut trees. We've already started the work on this part of the project, and this slide shows our progress to date, where we're engineering chestnuts with the ubiquitin promoter-driven OXO gene. We've already produced over 100 PCR-positive ubiquitin OXO transgenic events in 9 of the different founder lines, and these lines, of course, represent all the regions of the American chestnut range, so we're happy about that. This shows the plasmid that we're using, and you can see up here it shows the ubiquitin 11 promoter driving the OXO gene. These are the PCR results, one of the PCR results. These 3 lanes here show chestnut transgenic events, and they show the OXO gene is present there. This lane right here is the plasmid itself, so it's the positive control, and these other lanes are like water and empty, so they shouldn't be signal there. Finally, we've already gone on and produced our first somatic embryos from multiple PCR positive events from this work, and our first somatic seedlings are germinating. They may look pretty ugly, but this one looks pretty promising. It's got a wonderful taproot on it, and we hope that we'll get lots of plants eventually from these transgenic events. This particular event is in a Vermont tree background. It's another one of the founder lines. Work on our second new transgenic approach is in the planning stages still. Jared and Jason Holliday at Virginia Tech have identified chestnut genes that are strongly induced by blight infection. We will use the promoters from these genes to drive the expression of OXO. For this work, we actually plan to collaborate with one of the top university plant transformation labs in the country, which has decades of experience building the kind of DNA vectors we plan to use. And this lab just happens to be right here at UGA. The third new approach we will test with OXO gene is also still in the planning stages. It uses tissue-specific promoters to drive the OXO gene. In this case, we want to use promoters that strongly turn on genes in the inner bark of the chestnut tree where blight infection occurs. This photo shows the GUS gene driven by an inner bark tissue-specific promoter only turns on GUS expression in the inner bark of a transgenic citrus tree stem. We will use promoters like this one to turn on the OXO gene in the same tissues that show GUS expression here, and in chestnut tree instead of citrus, of course. Now, there is accumulating evidence that OXO alone is not likely to provide full resistance to the chestnut blight fungus. Bruce Levine will give more detail on what's shown in this slide later today, but it shows the results of a small stem assay conducted by Bruce using mutant lines of the blight fungus in which the gene that produces oxalic acid has been knocked out. So they can't produce oxalic acid. And you can see the cankers here. These are the mutant lines, and these still produce cankers, even though the fungus in these lines doesn't produce oxalic acid. So that's the message. Even without oxalic acid, the blight fungus still induces cankers in American chestnut. So what are we going to do about that? Well, this is the last of the strategies I'll talk about today. It's called host-induced gene silencing. And if we have to rely on other genes besides OXO, this would be a good way to go. We want to engineer chestnut trees so that they actually silence specific genes in the fungus, making it less virulent. On the right here is an example of some work from Angus Daw, and it shows that knocking out a single gene in the fungus makes it grow more slowly, which would make it less virulent. The cartoon on the right actually shows how a transgenic approach called RNAi can be used to silence specific genes. So we might be able to make a less virulent fungus by engineering the tree with these genes. Details of how this actually works, which are shown in this left-hand panel here, are for another day. But this approach has been successfully used to give disease resistance to other plants. Well, thanks for your attention. And I'm sure Jared will do a great job of answering your questions, and I hope to see you all again soon.
Hey everyone, my name is Jason Holliday. I'm a professor in the Department of Forest Resources and Environmental Conservation at Virginia Tech. I've been working with the Chestnut Foundation for about 10 years now on a variety of projects, previously mainly related to the back cross breeding program. And a couple of years ago, Jared Westbrook and I decided we wanted to have a better understanding of what makes Asian Castanea species resistant to the blight. And use that information, and if we can discover that in those genes that are creating resistance in Asian Castanea species, maybe we can use them to improve American chestnut. And this is an approach that some would term cisgenics, where we're taking genes from the target species, i.e. American chestnut, or a closely related species, e.g. Chinese chestnut, and using those to confer resistance to our target species. And ultimately, maybe the goal would be to have one of the OXO constructs that Scott was talking about, where we have OXO, but it's driven by a different promoter than the one that's used in the Darling 54-58 tree. And then in addition, one of the blight resistance genes that we identify through our work, and then also a gene for phytophthora resistance, which is also a critically important disease pressure, especially with climate change. How do we go about doing that? Well, if we're working a crop plant, it'd be a lot easier. But since we have a tree and it takes a long time to generate crosses and progeny and so on, we have to kind of adapt. And so we do have a lot of interesting resources. One of the resources is the back cross breeding program itself, where we have these large populations of trees that have the result over multiple generations, have resulted in a population that varies in its blight resistance. We can use that information to do what's called quantitative trait locus mapping or genome-wide association studies. Basically, what that means is looking for the parts of the genome in these populations that is driving the resistance that we see. So some trees are resistant, some aren't. What are the regions of the genome that underlie that? What that gets us to is a large number of candidate genes in those regions, some of which have nothing to do with blight resistance, but some of which do. So that's one item on the list. What we find from that effort is that there's a lot of locations in the genome that are related or correlated with blight resistance in the back cross breeding program. And so this is just an illustration that Jared Westbrook made, showing where those places in the genome are in the different populations that TACF works with. So we have clapper and graves, which are two of the main foci of the breeding program, and then a few other possible populations where we can identify these regions. And the colored bars are just where are the genes, or where are the chromosome regions that are conferring blight resistance. And you can see they're kind of scattered all over the place, and they vary a little bit depending on what your source of resistance is. So what's the Chinese great grandparent of each of these populations? So that gets us to thousands of genes. Another thing that we can do is look at what genes get turned on in American chestnut and in Chinese chestnut in response to blight infection. So we did what's called an RNA-seq experiment, where we basically, in collaboration with Hill-Craddock and some others, did this infection of the trees, and then looked at which genes are turned on and which genes are turned off. And we're specifically interested in the genes that get turned on in Chinese chestnut, but not American chestnut. Those would be the ones that are likely to be involved in conferring resistance in Chinese that does not exist in American chestnut. So that's a list of several hundreds of genes. Another thing that we can do is look in Asia at all the Asian Castanea species and ask the question, what parts of the genome has natural selection targeted in the wild over millions of years, and add those regions or those genes to this list. So there'll be lots of places in the genome that have signatures of natural selection that we can identify. Many of those, or most of those, will have nothing to do with blight, but because the Castanea species in Asia co-evolved with Cryphonectria, the blight fungus, presumably there will be some signature of the adaptation that those Asian species have experienced over the past millions of years, to enable them to be resistant to the blight fungus in the wild. And so what we ultimately want to do is take those different lines of evidence. So we have regions in the genome from the breeding program that appear to be related to blight resistance. We have genes that are turned on in Chinese chestnut in response to blight infection, and we have genes that seem to have been targeted by natural selection in the wild in Asia. And if we kind of overlap all of these lists, we get down to a relatively sort of tractable number of genes that we can go forward and test in different ways in order to understand their role and their possible usefulness in conferring blight resistance in American chestnut through the development of additional transgenic or genetically modified trees. And so this figure is just showing the overlap between those different lines of evidence. So the bar chart on the top right and the list of items in below that is just what happens when you require each of the different quantities to be true. So it needs to be for example, it needs to be in a quantitative trait locus. It needs to be increasing in Chinese chestnut in response to blight, or it needs to be a target of natural selection. How many genes are left when you require all three of those things to be true for a particular gene that we're interested in? And what we end up with is maybe 50 candidates that look promising and look like they're worth additional effort to understand if they can help with blight resistance. And among those genes, there's lots of different biological processes that are happening, things like pathogen perception, the transmission of the perception of the pathogen downstream in the cell, possibly some are related to apoptosis or programmed cell death, where the plant will kind of wall off a bit of its tissue so that it doesn't, so that it can kind of prevent further infection or defensive metabolites and strengthening of the cell wall against the pathogen. So we would like to probably target the genes that are involved in the most upstream portion of that, which is the perception of the pathogen, so it doesn't trigger this cascade that might be happening in American in one way and Chinese in a different way. And so we're working with John McDowell here at Virginia Tech and Amy Bruner, both professors here, to try to develop ways to test these candidate genes without going through the significant effort of making a genetically modified tree for every one of them, which takes years. And so one thing that John McDowell is working on is this so-called VIGS system, where basically you can infect a leaf with a virus that carries a gene that you're interested in, and selectively turn that gene off. Okay, so we would want to do that in Chinese chestnut, for example. If we find that this gene is related to resistance in Chinese chestnut, what happens if we turn it off in Chinese chestnut and then infect it with blight? If Chinese chestnut subsequently gets blight in a way that it doesn't when we don't turn off this gene, hopefully that wasn't too many negatives to understand, then this gene might be something that is worth looking into further. And so going from a list of 50 down to a relatively smaller list that Amy Bruner here can then put into new American chestnut lines and actually make transgenic or genetically modified trees, and test the effectiveness of these new candidate genes in the greenhouse and ultimately in the field.
Thanks, Jason. My name is Bruce Levine. I'm at the University of Maryland, where I am a perpetual PhD student, and I'm working on the fungal side of the equation, looking at the host pathogen interaction, very much the same kind of approaches that Jason and Scott talked about, but working on it from the fungal side. It can also help with gene discovery. It can help us understand the host pathogen relationship better. What does a fungus do to the tree? And how is that different in susceptible Americans versus resistant Chinese? That can lead us to understand what genes in the trees are responsible for susceptibility or resistance. The advantage on the fungal side is it's a lot easier. We have well-established methods for genetically modifying this fungus. Instead of taking 2 years, as Scott mentioned, from cell to having a transgenic plant, you can do it in a matter of weeks with the fungus, and it's pretty efficient. It's pretty reliable, and it's getting better through the use of CRISPR. So it allows for us to do a high throughput analysis of fungal genes to see which ones are important in the process of pathogenicity. What are they doing to the tree, and are they worth pursuing further? Or if they don't make a big difference, then you skip them and move on to the next. The OXO trees like Darling 58 or Darling 54 are actually an early example of success from fungal genetics. Back in the 1980s, they discovered that when the Cryphonectria parasitica, the chestnut blight fungus that I will henceforth call CP, when it was infected by a virus that makes it less virulent, the fungus stopped secreting oxalic acid. And they knew oxalic acid was involved in other pathosystems. So they said, this is probably important. Eventually, they found the gene that was responsible for the synthesis of oxalic acid in CP. It's called OAH1. And eventually, they created knockout strains of the fungus with OAH1 deleted, inserted that into chestnut. And you can see the pictures on the right. They got much smaller cankers. So what you're going to see in a second is I got a hold of some of these OAH1 knockout strains from the Agricultural Research Service. And I did a small stem assay with them, as we now use for our inoculations, compared to a wild type strain here called DK80, which is fully virulent. And the red bars are the size of the canker at 45 days. So on the left here, this is DK80 at 45 days, big cankers. The two knockout strains are in the middle. And you can see the cankers are both much smaller. But off on the right is another fungus I used called Sclerotinia. So what the measurement of these cankers shows is that you get big cankers with the wild type fungus. With the OAH1 knockout, you get significantly smaller cankers on average. But then I took this other fungus called Sclerotinia, which is famous for producing lots of oxalic acid, and inoculated that. It was not known to be a chestnut pathogen. I inoculated it into chestnuts. It did make cankers, but they were very small, and they didn't really grow much between 19 days and 45 days. And so what these are telling us is that although oxalic acid is important, it's not the only story. You take it out, yes, the fungus is less virulent, but these results are also showing us that they're not non-virulent. The pathogen can still cause disease. And you have bigger cankers when you inoculate the trees with CP that has no oxalic acid than you get when you inoculate it with Sclerotinia, which has tons of oxalic acid. So there are other mechanisms of pathogenicity involved. Okay, you saw this picture earlier, and Scott explained what it's telling you. Just take a look at these circled cankers. These are pretty big cankers. And one of them, and the one in the middle, even killed the seedling within 45 days. So, we need to be looking beyond just the OXO mechanism. As great an accomplishment as it was, there are other things to discover that are, which are separate and independent of oxalic acid. Brings us to the question of which fungal genes should we study. And this is the part where up till a few years ago, we didn't have a lot of to go by. There's, like 10,000 genes in the fungus. Which ones should we knock out? Most of the work done to date where they knock genes out, and the list you see is only a partial list. There's a couple dozen probably. And where it resulted in reduced virulence, it did so because it really just weakened the fungus. Even in a petri dish, the fungus didn't grow well. They disrupted the fungus's fitness or its ability to adapt to life in the host. And maybe those kinds of genes will be good targets for host-induced gene silencing, RNAi, that Scott talked about earlier. They may very well be, especially if you go after a few genes at the same time. But they're not telling us much about the host pathogen relationship. They're not the fruitful ones that we need to go after. But in the last few years, there have been recent discoveries about necrotrophic pathogens like CP that give us some better choices. A necrotrophic pathogen is a pathogen that kills the tissue of the host that it's in, and then feeds on the dead tissue. And that's what CP is. It used to be thought that it was all brute force, that the pathogens produced toxins or they produced enzymes that dissolved tissue, and that's how they got in there and grew. But we now know that brute force is not the whole story. Sclerotinia has all of those things, and it's just not really doing super well in these chestnuts. There's an additional factor that's emerged in importance called effector proteins. And effectors are proteins that are encoded by genes. They tend to be very small. They tend not to look like any other known proteins. And the definition is that they are little proteins that interfere with the defense functions of the host. So that's why I have this graphic of the alarm keypad. You can think of them as the molecular equivalent of knowing the alarm code to turn off the defenses of the host. And that's why highly adapted pathogens like CP are so virulent against chestnut and Sclerotinia is not, because it doesn't have the codes for the chestnut. For this year, I have my sights set on 5 or 6 genes to knock out. And going back, I'm going to start with one called NLP1, which is a toxin. And NLP1 is common in plant pathogenic systems. And it is produced by CP. And the interesting thing about it is some plants, like Arabidopsis, have proteins that are sensitive to NLP1 and act as defense signaling molecules. So the toxin gets into the plant and the plant knows that it's there, and instead of getting sick, it increases its defenses. So we want to see if NLP1 is important. If we delete it from the fungus, does the fungus get less virulent? If so, we'll look in Chinese chestnut to see, does it have genes that are sensing NLP1 that American chestnut doesn't have? And even if we don't find them within the chestnut family, maybe we can move them in from Arabidopsis or another plant and see if they work in chestnut. The next set of genes I plan to target are polygalacturonase genes. There are 2 of them in CP. Polygalacturonase is an enzyme that dissolves pectin, which is what holds plant cell walls together. Previous work done in the 90s found that Chinese chestnut bark extracts were 15 times more inhibitory of polygalacturonases from CP than bark extracts from American chestnut. And so Chinese chestnut does seem to be blocking selectively the action of these enzymes. So let's take these enzymes out of the fungus and see how big a difference it makes. If it's significant, this is a very fruitful area of further research. So the next slide, this is going to be about looking for effector proteins. And this is sort of the newest area of interest. Some work was done in Switzerland where they examined the profile of effectors found in CP. So this rather confusing chart, but I'll explain it. The horizontal lines each represent a different fungal sample. The lime green color are all different isolates of CP. And then the other colors are different fungi that are closely related to CP, but which are not necessarily virulent pathogens of chestnut. And the vertical columns, each one represents a different protein or a different effector protein. And you can see that some of them have very close homologs in other fungal species. And as you go across, you can see they're found in many species, but there are 3 here which are unique to CP, which is also uniquely virulent in chestnut. So these seem like very good candidates to knock out. If we see a big reduction in virulence, it would be very much worthwhile to follow up and see what these effectors are interacting with in the tree. It will certainly lead us to some important information. So this gene knockout method, we're not going to discover everything there is to need to know. But with this approach, we can rapidly discover maybe a few useful things that we can follow up on. We don't need to know everything to improve to make new discoveries that will lead to more resistant trees. So that's how this line of work contributes to that effort. Thanks.
As you guys have been listening for the past hour or so, our science program is pretty busy. We've got a lot going on, a lot of irons in the fire, a lot of projects that we're working towards. But you probably gathered from the presentations today that a lot of those projects are fairly early. You know, they're still happening with our research collaborators. They're happening in laboratories, and in, you know, in research locations, but they're not really something we can get our hands on yet. And that's, you know, getting our hands on things is where our citizen science program really, you know, kind of shines. That said, I think the biggest questions we've been getting lately have been, okay, well, so what are we doing? And we have plenty to do in the coming season and in the years to come. The science coordinators and outreach coordinators have been working to develop a planning template that we're going to share with the chapters. But anyway, within that planning process, you know, we're really looking to identify not only scientific priorities that our chapters and volunteers can help with, but also things that are going to be beneficial to our chapters. So I'm only going to focus on the kind of hands-on field activities for today. But for those of you that are active with your chapter, be prepared. We're also going to be talking with you all about some more outreach and you know, kind of chapter management projects that we could be working on to really just sort of strengthen our chapter program and build out the kind of core of workers and volunteers that we have in our programs. So the first thing I want to talk about is germplasm conservation. If you've been following our Chestnut Chats and following our organization, this is something we've been working on and we will continue to prioritize. You know, a lot of the projects that we heard about today would really benefit from diversification. You know, once we've gotten to the point where we have a new product that has reliable, robust resistance, and we want to get that out into the forest, we need a population of trees to cross those into. So continuing these germplasm conservation efforts is really important within our chapter network. So, you know, continue finding wild trees and planting germplasm conservation orchards or GCOs. Jared also talked a little bit about the screening we've been doing of our backcross material. We found, you know, dozens of good trees with decent levels of resistance, but that also means that there are a lot more than dozens of trees that haven't really shown to have great levels of blight resistance, but they are highly American and they represent wild American chestnut germplasm that we have captured into our orchards, and we'd like to maintain that. So, you know, maintaining germplasm conservation orchards, maybe developing and maintaining backcross conservation orchards. You know, that could be maintaining the trees right where they are. But if you know, landowners want to reclaim some of these backcross orchard locations, we can still get those trees collected into more of a consolidated space. It's actually, we've always found it really useful to have collections of trees that we can work with kind of en masse in one place. It makes our work a lot more efficient. So those are some great places to focus. For those chapters who have really hit germplasm conservation pretty hard already, you could certainly work with your science coordinators to take a more targeted approach and try to figure out which parts of your state or unique ecotypes maybe haven't been represented, and focus on finding those more rare sources that are worth keeping in your collections. Jared hit on this right at the beginning, the Best by Best program, and this is kind of the evolution of our backcross program. You know, as you know, I think this is probably the best example we have in our programs of adaptive management. You know, we had this very linear backcross program we were working towards for a long time. We learned some things about how resistance was inherited, how many genes we were trying to find, how backcrossing was working, and we made adjustments. And those adjustments have allowed us to find some trees from the backcross program that do have high levels of blight resistance. And so our chapters can really work on making those best by best crosses. That's a great focus for the coming year and probably the next several, you know, few years at least. You know, the trees trees don't go anywhere, but they still don't always cooperate when you're ready to make crosses on them. They may not be flowering well. Access may be difficult for whatever reason. We had a frost this year that got in the way of a lot of the work we were planning to do in some locations. We just didn't have the flowers available. So making these best by best crosses, doing priority pollen collections over the next few years so that we can freeze pollen and really pinpoint specific crosses we want to make. Jared kind of went over the rankings that we're doing within our backcross program of selections that are, you know, the very best are our blight 1 and the next best are our blight 2. We're also doing those same kind of assessments for trees with regards to Phytophthora root rot resistance, or PRR. So the PRR1 and PRR2 trees are high priority for Phytophthora resistance, and those are also great trees that we can collect pollen from, and those exist kind of throughout the whole range of the chestnut, the whole range of our chapters. And then finally, starting to develop regionalized best by best orchards. These are really kind of the next step seed orchards that our chapters can put in and start trying to produce some improved seed from the Best by Best program. Kind of following along with Phytophthora root rot, this is another area that we could really use some more focus on. Our southern chapters have been working on this for a number of years now. I would say at least 15, maybe closer to 20. But that buy-in, I shouldn't say buy-in, but the activity with breeding for Phytophthora root rot resistance has been primarily in the South. And this is an area that we could certainly see more participation from our northern and sort of central range chapters. There's still material that we can screen. We have a great partnership with the U.S. Forest Service at the Bed Creek Experimental Forest, and we can continue to send them material to screen and identify, you know, particular individuals that have higher levels of Phytophthora root rot resistance. And we can also start establishing more Phytophthora root rot resistance orchards. In the South, we want those to be with, you know, utilizing trees that have really high levels of Phytophthora root rot resistance, but we could also plant in locations without Phytophthora, you know, farther north, trees that maybe have intermediate levels of resistance, but if we can cross those together in those locations, we could produce seed that could be sent to places that have more issues with Phytophthora. Because certainly as climates warm, we expect Phytophthora to expand its range farther north and become more of an issue for more of the native range. But it's certainly an issue already for a good chunk of the folks working with American chestnut. Between the Best by Best program, the Phytophthora program, and germplasm conservation, knowing how to make controlled pollinations is a skill that's really valuable for our members to know how to do. And so if this is something that you don't know how to do and you'd like to learn, it would be a great, this coming season would be a great time to learn how to make controlled pollinations of American chestnuts. It's helpful for pinpointing the correct time to collect pollen and also for actually making particular crosses of interest. So if this is something that you'd like to learn, I'd encourage you to reach out to your science coordinator, your outreach coordinator, your local chapter, and see what pollination plans they have in the coming season. And if there's a way that you can get involved and maybe learn from someone that has already, you know, that has experience, that knows how to do this particular task. It's my favorite part, honestly, of our field work is pollinations. I just think it's a really neat thing to get to do. And the more people that know how to do it, the better. Finally, I wanted to talk a little bit about reintroduction trials, not necessarily to encourage a lot of these, but to let y'all know that this is something else that, that we can certainly consider putting our efforts towards in the field. We've done a lot of restoration trials over, or I shouldn't say restoration, reintroduction trials over the years to start figuring out best management practices for getting chestnuts back in the forest. Often, you know, some of these we've done with wild type Americans, some of these we've done with our best material, you know, whatever was the most resistant at the time, to start testing these things on the landscape. Typically what we're looking for in a reintroduction trial is a large planting. I think the minimum size is about 300 nuts or 300 trees, but typically we're looking at 500 to 1,000, so a few acres. good protection, and we're looking for hosts that can maintain these things long term and collect data and report back. So these aren't necessarily for the faint of heart. These are pretty big projects. And so we've done a lot of these with state and federal forestry partners and other landowning partners. But these are really great projects to get underway. If we have something that we want to follow long term, these reintroduction trials are a great way to get material in the ground with someone who will do that follow up and see how our trees do 10, 15, 20 years down the road. Finally, I wanted to talk a little bit about sort of the adaptive management approach that we take with a lot of these projects. And again, I think the backcross program is a great example of this. We had this very linear path that we thought was going to work with the backcross program. But as we kind of worked through evaluating the trees that we were producing at various steps along the way, we realized that the resistance levels weren't what we expected them to be. And so then we were able to make some adjustments and implement those adjustments to then, basically transform our backcross program into now our best by best program. And these kind of iterative shifts are pretty common and also just good practice. We want to make sure that we're keeping an eye on the things that we're doing and assessing them as we go to ensure that we're putting our best effort in the best places possible, and that the effort we're putting in is actually going to produce something useful. So I would expect some of the projects that we presented today may shift a little bit by the time we get them to a point that we could use help testing them in the field. But again, that field testing is, and kind of proof of concept piece is a great place to start pulling our network in once these projects get off the ground to the point where we're ready to do that. I think that's my last slide. And Sarah, I'll turn it over to you.
Thanks, Kendra, and I appreciate you pulling that through. On my connection here in Indiana is a little unstable. Hopefully, I can keep it here for the last bit. Again, just showing another slide from last session, and what I love about the American Chestnut Foundation, what I love about hosting these and working with all these projects is how many people we get to involve in all of these programs. So, Kendra, thanks for kind of bringing that all together. We'll end here and take some questions. We've only got about 20 minutes. We can just go ahead and end here, and maybe Bruce, Jason, Kendra, Vasily, if you guys can come on screen here so we can ask some questions. While I was going off screen and coming back on, I lost a lot of the Q&A. Kendra, are there any in there that you are of particular interest? While you find the older ones that I lost, I think I'm gonna, there are a couple in here that I see now that I want to grab. Fred Behringer asks, have the LSAs been crossed? And if so, what do the progeny look like? And I can address that. LSAs have been crossed. In some cases, they look better. In some cases, they don't. I know in some of the best back cross material comes from a combination of back cross trees with the LSAs. So some of that resistance does appear to be heritable. Jason, you unmuted. It sounds like you have something to say about it.
I was just looking at the data that Jared has for that, and I think the progeny of LSAs are about a little better than the average progeny is a little better than the average American. So about 15% on that 0 to 100 scale. That's the average. Some are better than that, and many are not. But so they're not as good as the average B3. As the average selected B3F2 tree.
Okay, well, that's interesting. But it sounds like we can pyramid. I mean, kind of what we've been discussing. And there was another question in there, and I can't see it right off the bat. But someone said, well, what's the best path? Is it transgenics or is it backcross? And it's not this one or that one. It's all of the above. And I think really what we're trying to get at is and I think what TACF has really said, even in the beginning of embracing transgenic technology, is none of these are silver bullets. Somebody wrote a silver BB, and I kind of like that. Really, it's going to be a combination of all of these techniques working together. A lot of these different resistance, the resistance that's inherent in the large surviving Americans, the resistance that we've been able to capture in backcrossing, the resistance that is conferred by OXO, the resistance that comes from some of the genes that Jason and his crew are looking at. You put all those together, and you're going to get a robust long-term population of disease-resistant American chestnuts. It's not something that we can deploy now or even in the next 10 years. It's going to be an ongoing. We establish our best back crosses now. We establish the best back cross times LSAs in the next couple of years. We establish the best transgenic lines crossed with those trees, and then you deploy those. So it's really going to be that iterative, ongoing deployment that'll ultimately lead to chestnut restoration. But again, we have to have the long view there. Kendra, did you find any good questions?
Yeah, well, I'm looking at the ones that folks flagged to answer live. Steve Black wants to know, what evidence do we have that the traditional breeding will yield stable resistance in an open pollinated scenario?
I'm happy to take that one unless Priscilla, you want it. Or Jason. No. I mean, I think that the issue with that, what evidence do we have? I mean, I think there's not a whole lot of long-term evidence because these trees have not been in the landscape a very long time. But I think what we can say is, as we cross more of these trees, we see enhanced resistance. As we cross them together, we see enhanced resistance. So I think that there is pretty good evidence based on the progeny in the past and what we're seeing currently that it should remain stable, but we don't have any direct evidence in the field. So I know Stacy Clark's on here. Hi, Stacy. She recently published a paper on the trees that she planted back in 2008, 2009. And those were the very first trees that came out of the backcross breeding program, and their resistance is higher than American chestnut, but still not quite what we would like to see in a quote unquote restoration population. But I think what we want to, what needs to be understood from that is those were the very first trees that were coming out from open pollinated trees and not selected. So from open pollinated, unselected populations. Now that we've done a huge amount of selection and removed 99% of the trees, we're seeing really large gains in the progeny of these trees. Those plantings are now going on in the reintroduction trials that Kendra mentioned. And so we'll be able to see the results of that now in 10, 15 years to really get to that heart of that question, Stephen, about, what evidence do we have? I think we've got pretty good evidence that it should remain stable, but the rubber will really hit the road once those trees have been in the forest for 10, 15, 20 years. Jason, did you have anything to add on that? I saw you unmuted.
Well, I just wasn't sure if the question was about, are the trees going to survive for 50 years or are they going to survive and their progeny for 500 years? And is it a question of, is the blight going to overcome the resistance factors and all that? But I mean, yeah, it remains to be seen, but it's an extremely strong selection pressure. And also because resistance in the backcross trees involves a lot of different factors in the genome, it's going to be harder for the fungus to overcome all of those sort of contributing genes that are going to be present. And also it's going to be creating an opportunity for additional evolutionary action to happen over the generations as these trees continue.
I see a question in here about, it sounds like the new approach will be like the ACCF program by crossing the best of the best. So for those of you who don't know, ACCF is the American Chestnut Cooperatives Foundation, and they've largely been using a very small population of large surviving Americans, or LSAs, like we showed earlier, the Ork tree, the Walbridge tree, the Amherst tree. Actually, I don't think all of those are in the ACCF program. But that's trying to take an inherent resistance. So Jason mentioned what he's seeing is about a not 15% increase, but it's on the scale from 0 to 100.
15 points. Yeah, I don't know.
15 points. I like that. On that scale is where those LSAs are, and we see some enhanced resistance in those LSAs. With the best by best in the backcross, I'd say, I think Jared mentioned, well, we're about 50 right now. So if Americans are 0, the LSAs are around 15. The best back crosses are around 50. From just the best back crosses, we can get to 75, maybe even 80. We don't yet know what we can get if we pyramid those together. We might be able to get even higher. But we start to think about these point scales, and then if Chinese chestnut is 100, when do we get to 100? Maybe you throw OXO in there and you get to 100. We don't know yet. We haven't been able to look at those just yet.
All right, Sarah, I got a couple that were flagged to answer live.
Okay, well, and
related.
looks like Vasily might have had something to say comment on
I'm
that
sorry.
too.
Just related to that same question that you addressed, Sarah, I just wanted to reiterate that the best by best improvement efforts on the original backcross program, that's not new, that's not a new project, so the improvement is ongoing. So that's already been underway, and there's a lot of progress been made already. So it's not a entirely fresh start. It's something that we're continuing to improve. But yeah, you answered the bulk of it.
All right, I've got a couple questions kind of related to resistance and how we select those. So what criteria are we using for genomic selection? What genes? And are you guaranteed better resistance to blight if both parents show such resistance?
Ethan, I think
Those
that's
two different questions, but they seem related. So
I mean, for genomic prediction, basically, I mean, it's a little bit hard to explain in 30 seconds, but we're basically taking the whole genome as a predictor of blight resistance. So we can build a statistical model that relates the genome sequence to the blight resistance index. And then we can take trees for which we don't have a blight resistance index, sequence their genomes, not the entire genome, but enough of it, and then use that as a prediction of the resistance in the absence of field data. And so it's not like which genes, it's like taking all of the genes all at once. So that's the best way I can explain it briefly.
Well, I think, Kendra, your second question, there's a couple questions here in the chat that keep coming up about somebody asked if resistance is binary, it either is resistant or is not, and they said, well, how can you have intermediate resistance if it is or is not? And that's just the point. It's not binary. There is this sliding scale. Again, if you have 0 as American and 100 as Chinese, and that's still kind of fake, quote unquote, from a quantification of really what resistance is. But let's try and conceptualize it on that scale. You can have anything in between, from 0 to 100. So it's not is or is not. It really is everything in between. And somebody else asked about the environmental effects. Environmental effects play a huge part in what resistance ultimately looks like. So those of you familiar with the disease triangle, it takes the host, the pathogen, and the environment to present disease. So, if you have a highly resistant tree in a happy environment, they will perform better than that same tree with the same genetics in a poor environment. And that's that environmental, that genotype times environment effect. Kendra, there's a couple questions in here for Bruce too. And Bruce, several people really enjoyed your I want to throw on the kudos from folks in the chat. They really enjoyed your presentation. Mike says, are there any significant differences between various strains of Cryphonectria parasitica? Might some do one thing to dentata and others do something else? Or is Cryphonectria's activity pretty consistent across various strains?
Yeah, that's a good question. So a lot of the strains were tested in the 70s and 80s, mostly in Connecticut. And we have a standard strain we use called EP155, which is considered very virulent. And then there's a standard less virulent strain called SG23. And they're pretty reliable in terms of producing bigger or smaller cankers, but they're all pathogenic. And there's a lot of variation from strain to strain. Why that is, we don't really know yet. But that profile of effectors is a little bit different from strain to strain too. So they may be, missing a couple things, or they may be making more of some toxin or less of some toxin in the tree, or whatever it may be. So there's some variation, but this is a fungus that spent a lot of time making chestnut trees sick. That's its specialty. So they're all pretty hard on chestnuts.
All right, thanks, Bruce. Stacy asks, how did the problem with mislabeling the Darling line not get recognized until recently? And I think this has been a good question that a lot of people have been asking. You know, how could you not know? And I think there was no reason to suspect there was an issue with that. And I think it wasn't until there was some evidence that there was reason to suspect it that it was followed up on. So, you know, I think another thing that I've gotten a lot of reaction to with TACF's decision from December is, you know, what have you learned? And I think we've learned a ton, you know, both in terms of scientifically, but also in terms of good practice and what we're doing. So, you know, I think going back to adaptive management, going back to, you know, iterative processes, there's a lot of things that we've learned that we're going to implement to make sure that we can improve based on everything that we've learned and make this make what we have in the future even better. Kendra, anything else that really pops out at you here?
Oh, there's a whole bunch of stuff, but we only have a few minutes.
Yeah.
I'm going to pick on a couple of Carolyn's questions are pretty interesting. Hi, Carolyn. She had some questions that I hope maybe Bruce can address. Let's see. What are the broader effects of modified fungal genes, say, on beneficial fungi in the environment?
Modified fungal genes. Well, we're not putting any of this transgenic fungus, you know, where we take a gene out or possibly add a gene in. We're not putting any of that out into the wild. This is all being done not to produce some kind of modified fungus that we're going to release that will be less virulent somehow. It's just done for research and to figure out what the genes are that are important and what they do in the tree. And knocking them out of the fungus is the first step. Yeah, if you started putting transgenic fungus out in the wild, then there would be some issues with, you know, whether it will, you know, affect the mycorrhizal relationships or whether the tree would adapt to whatever you did to the fungus and then end up becoming, or you would end up selecting for more virulent fungus somehow. But we're not doing that.
Cool. All right. I'm not sure if any of you guys can answer because this is a question Clark directed actually at Dr. Merkel, but I think one of you guys might be able to handle this one. He wanted to know if he understood correctly that you get multiple events with each gene insertion.
Yes. Usually. If you want them.
All right.
I thought William Coder's question was really interesting about the Best by Best program versus the traditional breeding program, and maybe highlighting some of those differences.
Can you read it?
Best by Best seems reminiscent of the preceding breeding program is the use of genetic data that makes the Best by Best program more likely to improve blight resistance when the classic breeding did not.
Well, I think that's a good question for Jason.
Well, so B3F2 is open pollinated, right? Arise from open pollination. Am I correct in that? Vasily?
Yes, mostly.
Yeah, so this is different. The previous would have been best by whoever. Best by best is taking your best pollen and putting on your best ovule. So it is somewhat different. You're selecting both parents instead of just selecting one parent. And so yeah, and there was another question about, is it expected to, you know, guaranteed to yield an improvement in blight resistance? And I don't know what guaranteed, but I would say you're gonna get an increase in mean resistance. Some of the progeny are going to be worse than either parent, and some of them are going to be better. And the average is going to be better.
And then the trick is just to pick the better to breed amongst one another, and you pick the
you
better.
Take the next
and
one
better,
until
better.
And as long as as long as you continue to have genetic variation for the trait, which is blight resistance, you can continue to improve the progeny. It's just a question of also of what the trade-off is going to be for how much of the Chinese chestnut genome you pull. And I think Jared's simulations show that you can do it in such a way that you don't start pulling too much of the Chinese chestnut genome, which might have knock-on effects that we don't know if we get too much of that.
Yeah. Well, we're pretty much coming here at the end. I'll do two more. One question was, do Chinese chestnuts ever get the blight? Absolutely. Chinese chestnuts can die of the blight too. So, and again, that's a factor of the genetics of the tree. It's a factor of where they're planted. It's a factor of other things that are affecting them. Like if you have a Chinese chestnut with lower than average resistance, and that happens, planted on a poor site that's been impacted and infested with Asian chestnut gall wasp, it's probably going to die from the blight. We see them a lot in shaded conditions die of blight too, because they can't handle the competition. So yeah, definitely that happens. Last question, and I'm not sure if anybody knows this, but so I'm going to throw it out to the group. Anita Klein asks, can you pick up on any epistatic interaction? Do we know that?
We haven't tried. I mean, I don't know exactly, in what context it's referring to, but that's pretty hard to do. I mean, from my perspective, from like a genomic perspective, I don't know if there's some other more focused kind of genetic perspective that this person's thinking of, but yeah.
Okay. Well, thank you all. I really appreciate you guys coming and sticking with us. Next month, we actually have another person from Virginia Tech, Joanna. Can you help pronounce her last name, Jason?
Not really. Malukowicz. It's a poet that I've never been able to pronounce accurately, probably.
Well, I'm excited to welcome her. Thanks for working with her, Jason. And we're going to welcome her. She's going to be talking about evolution of Castanea and Cryphonectria, what that interaction of the host and the pathogen can show us about how we want to improve these programs that we just talked about. So I hope you can join us in February for that Chestnut Chat. Looking forward to see you then. Vasily, Jason, Bruce, Kendra, and then Jamie, Sierra, Cassie, Lauren, Hoy. Thank you guys for helping do all the questions. Any final words for everyone before we take off?
I will say really quickly for folks who still have questions, some of these were addressed in our previous Chestnut Chats, so I would highly recommend going back and watching those. Our presenters did a fantastic job.
Thanks, Jamie. I appreciate that. And we're going to do our best to post these two in our archives. I saw in here that Hal is incredible with the video editing. So as long as I can get him the recording in time, he'll get that edited up. And he says that he'll get that up in the archives by Monday. So that's exciting too. And we got the YouTube to finally work. So those of you in YouTube, I hope that all went well. And yeah, so have a great weekend. Stay warm, everybody. And we'll look forward to seeing you in February. Thanks so much.